Okay, we're going to finish up our discussion of how things move through cell membranes today. We're going to go back to our big overall scheme of how this works. So if you recall when we first started this, we said that movement through membranes can happen one of two ways. It can happen through either passive transport or active transport. And we said that passive transport happens with no energy input from the cell. Everything always moves from high to low concentration. And the purpose of it is to equalize the concentration of the things both inside and outside of the cell. We've discussed both diffusion and osmosis, and we have one more piece of passive transport to discuss, and that is facilitated diffusion. And one way that you can actually think about this is that, well, if you think about your house, ants and spiders and even mice, they can all get into the house without you opening the door and inviting them in. They just seem to manage to find any little crack, any little crevice that they can to get inside of your house. You can even think about it the same way of diffusion. There's lots more ants and mice and spiders outside the house than there are inside the house, so they manage to find their way through without you expending any energy to let them in. They manage to find their way in. So that's kind of like diffusion. Facilitated diffusion, though, is what happens when your dog needs to get in. And maybe you have a doggy door cut into your door, and so the dog can go in and out as he pleases. It's not that he doesn't belong inside. He does belong inside if you let your dog inside. Obviously, if you cut a doggy door, you want him to come inside the house. And so he is able to go in and out of the house completely on his own whenever he chooses. If he uses the doggy door to get in and out. Sometimes the cat may choose to use the doggy door too if you happen to have one of those. But you have specifically made a way for the dog to get into the house through this special door that you have cut for him. Facilitated diffusion is kind of similar to that, because the cell wants things to be able to move in and out like water, but it can't get through the membrane all by itself. And part of the reason for that is that a water molecule is polar. And if you remember the way that the plasma membrane is set up, the outsides of it are hydrophilic, which like water, but the inside of that, where those fatty acid tails are, they're hydrophobic. They don't like water. They're opposed to water. And so the water can't just pass through the membrane because it can't get through that nonpolar middle. So in order for water, as just one example, to get through the membrane, you have to provide a way for it to get through the membrane. There are actually two ways for things to get through the membrane without having to require any energy input. And one is through a channel protein. And she has a picture of this in there where it's an integrated protein, an integral protein in the cell membrane, and it just kind of looks like a tube. It looks kind of like a slide in the way she's got her picture drawn, and the water molecule can just slide right through there without having to come in contact with all the hydrophobic parts of the cell membrane. So just kind of like your dog can slide in and out of the doggy door whenever he feels like it, the water can slide in and out through the channel proteins as well. Now, the author of your book, she doesn't say it in so many words, but she kind of seems to indicate that this is the only method for things to get in. That's actually not quite true. So I don't want you to be deceived there that channel proteins are the only way things can get in. You can also use what's called a transport protein, and it does not require any energy on the part of the cell either. And this is actually pictured on the bottom of the coloring sheet for passive transport that we were using the other day. So it's this one right here. And you can see that this protein right here has a spot in the middle that's got a specific shape that matches up with whatever it is that it's trying to import. And so, again, it's by the effects of concentration. So as these molecules are randomly moving outside here, one of them will kind of randomly go in here. The protein then will deform so that it will close on the one side and open on the other side, and then release that molecule inside of the cell. Again, that does not require any energy, and so it is considered a form of transport. And so transport proteins and channel proteins can both be used for facilitated diffusion. But again, the thing to remember is that facilitated diffusion just means they had to have a little help. Like the dog had to have a doggy door to get through. These guys had to have a little bit of help to get through the cell membrane. So now we're going to move to active transport. And active transport is just the opposite of passive, where we said passive transport requires no energy input. Active transport does require energy input. Here we're going from high to low concentration. Here we're going from low to high concentration. So if I'm going to move it against the concentration gradient, I'm going to have to utilize some energy in order to get that to come through the membrane. One way to think about that is if you think of a preschool teacher and she has let her 3 and 4 year olds outside for maybe recess, and then she wants to gather them all back inside. And so most of them are there, but she's got this one little boy, and he's outside and he's running around, and he has no desire to come back in because everybody else is inside. And so he wants to hang out where the concentration is low, because now there's no line for the slide, there's no line for the swings, and so he's a happy camper playing out where he is. If the teacher wants to get him inside, she's going to have to exert some energy to go out and get him and pick him up and bring him back inside with everybody else, because she's got to move him from low concentration to high concentration, okay? Same type of thing here. If we're going against the concentration gradient, energy has to be exerted in order for that to happen. Now, active transport will utilize one of 2 different methods to get things inside. One is the transport protein that we just talked about. And the method for these transport proteins to work will look exactly like it does on the bottom of your coloring sheet that we've already seen and that we've already gone through. The only difference is that for those particular types of proteins, energy is required to make them work. The diagram that's that's in your book where she shows you that, she has where it talks about ATP and ADP, and we haven't talked about any of those things yet, so that part of the diagram may not make a lot of sense to you. We're going to actually hit that kind of stuff in the next module. Basically, ATP is just the battery power for the cell. It's the energy source. It takes a molecule of ATP and turns it into a molecule of ADP with a whole bunch of energy. And that's how it powers anything that it needs to power in the cell that requires some sort of energy input. So that's all those letters mean. Don't worry about them right now. Just realize that what that signifies is that the cell is using energy to make that process happen. Okay, so that's transport proteins. The other thing that a cell can use for active transport are vesicles. And we talked a little bit about vesicles when you studied the different parts of the cell, the different organelles of the cell. And so vesicles are used very, very much in all kinds of cell transport. Okay, so there are 2 types of specific transport that vesicles are used for. So we'll divide this into 2 pieces. One is called endocytosis, and the other is called exocytosis. Now, endo, if you see the EN there, that's going to go in the cell. And exocytosis, X should remind you of the word exit, which means things are going to go out of the cell. Now, the cool thing about vesicles is that they are made out of the same types of phospholipids that the cell membrane is made out of. And so these vesicles are able to fuse with the cell membrane and detach from the cell membrane, and the cell membrane can go back together. Remember how we talked about how we said if the cell membrane gets interrupted, it very easily can reassemble itself? Well, this is exactly the type of situation I was talking about, except we weren't ready to talk about that quite yet. And so the cell membrane can reassemble itself once it has allowed things to go in and out of it. So if I think specifically of endocytosis, there are a couple of different types of endocytosis, and I'm going to do this in blue, not because the color means anything, but because I think my black's getting a little bit faint, and I want you to be able to read this. So again, the color doesn't mean anything. So endocytosis can be either phagocytosis, or it can be pinocytosis. Now, phagocytosis literally means cell eating. And so phagocytosis is used for molecules that are very large, large solid molecules, particles, or they may even be another whole cell. Amoeba do this a lot, so when we talk about amoeba, we'll hit this again. There's a picture on the bottom of page 171 where you can see different photographs in the series of an amoeba eating another type of cell. And so what it almost looks like is that the amoeba is like kind of extending its, kind of looks like arms is really kind of what it looks like, but it kind of pushes its cytoplasm out and makes extensions on the side of the cell, and it goes all the way around this thing that it's trying to engulf. And then when it does that, the cell membrane actually then has completely surrounded it, and it will kind of fold in, and it will separate off, and then it will form a vesicle around this piece that it has just taken in, the substance that it has just taken in. And again, because the edges of the vesicles and the cell membrane are made out of the same type of phospholipids, it's pretty easy for those to just break apart and then reassemble themselves. And then the vesicle will take that thing that it has just ingested to other places in the cell to do whatever it needs to do with it. Okay, so that's phagocytosis. Pinocytosis is what happens if it needs to ingest something that's dissolved. So pinocytosis is when it's going to ingest some fluid that has dissolved particles in it. So phagocytosis means it's ingesting something that's large and solid. Pinocytosis, it's when it's ingesting a fluid that's got something dissolved in it, salts or sugars or whatever other type of substance it may be that the cell happens to need. Okay. Similarly, exocytosis is the reverse of endocytosis, where you have a vacuole that contains something, and that vacuole, the vesicle. You have the vesicle that contains something. The vesicle is going to move to the edge of the cell. It's going to fuse with the cell membrane, and then it's going to open up, and then whatever's inside is going to be released. So exocytosis is going to be used whenever the cell is getting rid of waste. And remember, that can be either excretion or egestion, either one of those two. That's going to be used here. Or if the cell is going to be doing secretion, where it has made something that another cell needs, and it's going to excrete that out into the bloodstream or out into the extracellular fluid so that another cell can get a hold of it. That's going to be secretion. And again, a secretion vesicle would be used. Waste vesicles would be used for these. And so all of these types of things are going to use vesicles. And they're things that are too big to go in and out any other way. I mentioned, just as an analogy, I said, okay, diffusion and osmosis. I mean, diffusion is kind of like when you get ants and spiders that come into the house. Facilitated diffusion is when you cut a doggy door in your door so your dog can go in and out as he pleases. Both of those things don't require you to get up off the couch and open the door to let anybody in, okay? However, if your grandma comes and knocks on the door and she's coming for a visit and she has all her luggage, at that point, you're going to get up, you're going to go outside, you're going to help her carry her luggage in, you're going to open the door for her, and so you are actively exerting energy to get her into the house for her visit. Okay, so that's basically the kind of difference here. And all of these things, in endocytosis, that's the best example for grandma. In endocytosis, you want grandma to come in the house, you want her to visit, you want the cookies that she brought you, you want the gifts that she brought you, and so you're going to exert energy to get her inside of the house. The dogs can get in through their doggy door, so you don't have to exert any energy, you don't have to get off the couch. and go away from studying your biology to help the dog get in. The ants and the mice can get in all by themselves. They don't need any help, so they come in by themselves as well. So, again, it's not a perfect analogy because you don't have more grandmas outside than you do inside, so the concentration exactly doesn't work. But yeah, hopefully you get the point that you have to exert energy for one and not for the other. So that finishes up our discussion of movement through membranes, and we have now come to the end of the module.